Ladies and gentlemen, welcome to today's webinar about AFM applications for smart and functional material studies. Our today's lecturer is Stanislav Leesment. During this webinar, you are able to ask questions, and we will try to address these questions at the end of the presentation. And just in case, if we will not address some of the questions because of the time limits, we will then contact you by email. So, Stanislav, you're very welcome.
Stanislav, thank you very much. Good day, good evening, good morning. There are pretty many attendants today. So starting from Eastern Europe, finishing with US. It's very nice to see you all online today. So our today's topic is AFM applications for smart and functional material studies. So when I started to prepare this lecture, so my motivation was following. We are analyzing the papers which are done with the help of our instruments around the world. Actually, there are about 1,000 papers appearing annually. So what we saw, the tendency that more and more groups are switching to smart and functional materials. Another evidence of this fact we could see on multiple conferences which are held nowadays. So these are really hot topics. And if we're talking about material properties at nanoscale and nanomaterials, it is definitely necessary to have a good tool to make studies of small objects. So atomic force microscopy and scanning probe microscopy in general is one of the main tools for characterization at the nanoscale. And again, when I started to make the structure of this talk, it was pretty confusing because we have multiple kinds of applications, multiple kinds of properties, multiple kinds of samples, and various AFM techniques. So I got actually a three-dimensional graph, and which is really difficult to put into the plain presentation. So we decided that the best way for us, like for producers of the equipment, is to start from the point of view of techniques, the AFM and the SPM techniques. So, for those who are not too convenient with AFM, I will just shortly repeat how it works. So we have the sample which we are studying. We study it with a special probe. In AFM, this is the flexible cantilever with a sharp needle on the end. So with a laser, we go to the backside. We focus the laser on the backside of the cantilever and get the reflection to the photodiode. So when the probe bends, the reflected light moves along the photodiode, and we get the signal which is proportional to the bending of the cantilever. And the sample is fixed on the precision XYZ positioner, which is brought closer to the tip with a stepper motor or with some other system, and thus we can the tip starts to touch the sample, and thus we can record different properties localized by the tip. So this scheme is called sample scanning, and but in my presentation, most of examples will be done with the so-called tip scanning scheme. Actually, the idea is the same. The only thing is that in this case, the tip is connected to the XYZ positioner. And but from the point of view of tip sample interaction and from the point of view of properties we can probe with this scheme, it's actually absolutely the same. Just not to confuse you. So the first thing which people expect from AFM is that with AFM we can get the high resolution, which is not achievable, for example, in optical techniques, high resolution of the morphology of the sample, so we can study it. And the main three approaches how we can do this in atomic force microscopy are following. First is contact mode, when the tip is in static contact with the sample, and we keep the bending of the tip constant and profile the surface in this configuration. Another way is tapping mode, and when we oscillate the tip around the resonance frequency and keep the amplitude damping constant. And the third way, we call this hybrid mode, but it's actually based on the so-called jumping mode technique, when the microscope works as the sewing machine and the tip interacts with the sample in this way. So let's start with the first one. You could have heard different names of this technique. Scientifically, it's called amplitude modulated atomic force microscopy, but probably you have heard also the different names like tapping mode, or semi-contact mode, or intermittent contact mode, or AC mode, or non-contact mode. But actually, these names are about the same technique. So, and why we start with this technique? Because from our expertise, from our knowledge, about 90% of papers, when we see the topography of the sample published, so the papers published in the journals, they use this technique to get the distribution of the sample properties along the sample surface. And how it works, we oscillate the tip at the resonance frequency. When the tip comes closer to the sample, the amplitude is damped, and we are tracking this damping, keeping it constant, thus keeping the tip sample separation constant. And thus we can profile this the sample surface. I will not give too many examples of the morphological studies in semi-contact mode. Otherwise, it should be a good review for several hours. But I will present a few cases about this. So the first case you see on the slide, it concerns studies of self-assembling amyloid peptides, which formed fibrils, which actually cause bad thing, which is called amyloidosis, and which is the reason for Alzheimer and Parkinson diseases. So it is very important to understand the mechanism of fibrillogenesis, how these fibrils appear. So in this work, the AFM was used to see the morphology and to calculate the length of these fibrils, and thus the researchers could understand the mechanism of peptides bonding. Another in this work, the group from Israel, they were studying the DNA. So they studied the morphology of these molecules to verify the process of synthesis, and they were trying to use these DNA molecules as a framework on one-dimensional conductors. And then in this case, these DNAs were used as skeletons for the nanowires, for conducting nanowires, filled with the silver, and the morphology studies of the sample could explain how the silver bunches go inside this molecule. And in this work was dedicated to studies of sorry, there is wrong header of the slide. In this work, the studies of patterned hydrogel films were studied, and the study was dedicated to research how these films are influenced by different kinds of impacts. And on this particular image, you can see the AFM, the consequent AFM scans of this film after ultraviolet irradiation, and it was shown that the sizes of holes in this film could change along the length of the irradiation, decreasing sizes from micrometer scale to nanometer scale. So my previous examples were about getting just the morphology in amplitude modulated technique in tapping mode, but being so simple like in explanation, actually this is not very simple technique. So there are there is a set of certain parameters which should be carefully chosen by the operator of AFM in to get the right results. Otherwise, the images captured with this simple technique are getting distorted. And on this slide, you can see three most common distortions or artifacts of tapping mode images. So first one is so-called switching. So from the left side, you see the switching. These white dots, white areas are caused by switching effect. And from the right side, you can see what should have been appeared in the right case. The second artifact is so-called ringing. So when the PID regulator is over-regulated and it's being excited, and you can see parasitic excitation. So from the left side is the common case of this artifact, and from the right side is the topography with the right parameters. And the third case is so-called parachuting, when the tip detaches at the high point of the sample and parachutes back to the substrate. And these parachuting effects can be also commonly seen. So taking the opportunity, I want to say that in our systems we have implemented the intellectual Tronic software, which automatically sets all the parameters for operating in tapping mode, and ensures that you get the reliable and high quality result without spending hours with your samples. So the next technique I want to talk about is so-called atomic force spectroscopy. So let's see how it works. We have the tip, and we vary the distance between tip and the sample, probing it in different points. And depending on the mechanical and adhesive properties of the sample, we're getting different kinds of so-called force curves. And applying the algorithms and apparatus of contact mechanics, we can extract the quantitative data from this. So this is called quantitative nanomechanics. And like my following example, yes. So the example of application of atomic force spectroscopy will be shown on spider silk studies. So, but before going to the experimental section, I will make the small introduction of this work. So the strength of spider silk is 5 times stronger than a steel cable of the same weight. So if you make the same size steel cable, the spider silk will be 5 times stronger. So it's very important to understand the properties of this material at nano level to understand how we can synthesize such kind of materials. The size of silk ribbons is about 10 micrometer wide and only few nanometer thick. So obvious question was how the silk is harvested. So this work was done by a group of Professor Hannes Schnipp from University of William and Mary, and they just collect the spider silk from the Brown Recluse Spider. So it's poison. It's not deadly, but it's actually very unpleasant. Yes. So if somebody bites, some of them bites you, you can see them. So they just have the farm of about 100 of them, and then they are getting the silk directly from the spiders. So after acquiring the sample, they are studied in AFM. And in this particular work, the mechanical studies were done on a piece of spider silk stretched over the hole. So we can see there are two islands, and the silk was stretched over this hole. And the probe with a spherical tip was taken, and the force curves were taken directly on the silk, on the piece of silk. And as if you see this force curve, this multiple force curves, they show exactly the same shape, which means that the silk is absolutely elastic, so we cannot make the plastic deformation at least at this level of force scales. And the young modulus of this piece of silk was calculated to be around 21 gigapascals. And together with measuring, we can use the AFM tip also to modify the surface at nanoscale. So in another work of this group, they they took the diamond probe and cut the piece of silk with a high force, and thus they could study that the breaking strength of a single nanofibril is about 120 nanonewtons, and to study the internal structure of this silk. So the atomic force spectroscopy is the great technique to study the mechanical and adhesive properties of the samples, but the main disadvantage of this technique is that it is pretty slow. So to make the single force distance curve, it takes roughly like starting from 0.1 second up to 10 seconds, but still, if you are going at the rate of 10 seconds, it will take huge time to collect the map of 512 by 512. So we have developed the first imaging technique based on the force distance curve studies. We call this non-resonance oscillatory mode or the hybrid mode. So in hybrid mode, we vary the distance between tip and the sample. So it is shown on this graph with the dashed lines. And together with a simultaneous measurement of the force response. So we have distance, we have force, and can build the force curves in real-time mode with a high speed. Speed is about a few thousand force curves per second. And having the force curve, we can and applying the certain mathematics, we can calculate the mechanical properties, quantitative mechanical properties, adhesive properties, and of course, first of all, we are getting the topography of the sample, keeping the maximum, the constant, the maximum difference of maximum force minus the baseline. So on this short example, we can see the advantage of the hybrid mode. So on the left image, we can see the morphology of the blend of two polymers. We know that these are low density polyethylene and polystyrene, but we cannot tell from this image who is who, and we cannot understand the fine structure of these guys. But looking at the adhesion map and at E-modulus map, which was captured in real time with hybrid mode, we can certainly see that there are two regions, like the matrix and the spheres. And calculating Young's modulus, we see that blue here is the polystyrene. Sorry, it's a polyethylene. The matrix is polyethylene, which is about 200 megapascals, and the blue and the green is polystyrene, which is harder, which is about 3 gigapascals. And what's curious, we can see that there are blobs of polyethylene on the top of the polystyrene, which are softer, and also give the different contrast in adhesion map. So together with mechanical properties, we can also get the value from the morphological studies with the hybrid mode. In this example, we've done studies of tungsten disulfide samples in vacuum. So the aim was a bit different, and not only to get the topography, but on this slide we'll talk about the topography. So when the sample was put to vacuum, and it's possible to do in our Integra system, you can put the sample inside the measurement chamber, put the cover on, and still having the optical access, having the electrical access, you can pump it down and make the studies in low vacuum. So in this case, imaging in a tapping mode or in a amplitude modulation mode, you can see the image from the right side. You can see some structure, but it is very blurred because in vacuum the huge surface charge appeared on the sample. And it was not possible to get the good resolution in a tapping mode. In contact mode, it was also not possible because in contact mode, the structures were just cut by the tip. So the compromise in this case was a hybrid mode, where we can control the force with high precision, and we can we can extend the force impact more than in tapping mode, keeping at the same time the good lateral resolution. So, and as you see, the topography in hybrid mode looks pretty clear. So the next technique I will be talking about is so-called spreading resistance AFM or conductive AFM or C-AFM. Traditionally, it is based on contact mode. When we have the tip, we're in contact with sample, we apply a voltage between tip and the sample and measure the current. So the conductive areas show current, and non-conductive areas don't show current, so we can probe the conductive properties of the sample with the high with high resolution. And one of the examples of these studies in this paper, C-AFM was used to confirm the photoefficiency at the grain boundaries of the material which is used for solar cells, and it was shown that, as we see, on the left side we have the topography, and on the right side we have the distribution of current, and we see that the grain boundaries they really show the conductivity, which helped to understand that the material which was novelly developed within the non-vacuum process can be used to produce the material for solar cells, and these solar cells will be much cheaper. That's okay if we have some kind of hard material like ceramics, but what if our material is soft or is loosely attached to the sample? In this case, the probe will either cut it or move it. So we applied the approach of hybrid mode. So in hybrid mode, tip traveling along the surface gets out of the contact in every point. So we really decrease the lateral lateral interaction between tip and the sample, and thus we can protect our sample from being changed by the probe. But at the same time, when we can control all this measurement cycle, and thus we can apply the voltage between tip and the sample only in certain window, which is defined by in the user interface. And if we define it only during the contact and apply the voltage and measure current during the contact, thus we can do the conductivity mapping in this mode, and we call this hybrid C-AFM. And this is one of the examples of this kind of studies. So there were studies of conductive nanotubes, which were spread over the surface with the electrical contact. on it, and we applied the voltage to this contact, and we're studying the conductivity of these nanotubes. And you can see that only the tubes which are touching this contact can be seen in from the on the right image in the conductive map. And so, for example, this tube from the right bottom part is not seen here, and it's obviously not touching the contact here. And moreover, we can see the gradient of current along the tube, which is pretty expectable. In these two examples, there are more two examples of such kind of studies. So on the top two images, we were studying the morphology and electrical, conductive and mechanical properties of single and multi-wall nanotubes. So the morphology was done, morphology is seen on the left, and on the right side we see the conductivity map of these nanotubes, and together with the E modulus, and we can define the single wall nanotube and the multi-wall nanotube here. And on the bottom slides, the study was reported in this paper. It was study of nanotubes covered with peptides, and some of them showed the conductivity, and so some of them also we studied the Young's model. The Young's modulus was studied, and from these images we see that not all of these nanotubes are first of all conductive, and the second, not all of these nanotubes have peptides deposited on them. So the next section of my lecture is about big, pretty big class of smart materials. And by definition, smart materials, all the piezoelectric materials can be defined as smart materials. So to study piezoelectric materials, we are using Piezoresponse force microscopy, and it classically works again in contact mode. We have the sample, we apply alternative voltage, and when tip goes along areas with different polarization, we are getting the different amplitude and phase response. Thus, we can study the domain distribution in piezo materials. And for example, in this work, the studies of piezo material doped with niobium was done, and it was shown that the dose of dopant, with the increase of dopant, decreases the size of the nanodomains of the piezo domains. Again, that's okay if we have some ceramics which is hard and we can study it in contact mode, and we can study the PFM based on this contact mode. But what if we have something soft or something loosely attached to the surface? And again, here we use hybrid mode, and in hybrid mode, during the contact point, we apply the alternating voltage between tip and the sample, and collect vertical response and lateral response at the same time. And as the tip is detaching between the points, we at the same time we don't affect our samples very much. Thus, we can go with more difficult samples. And here, like in 2010, it was reported that the diphenylalanine peptide nanotubes have the module, the piezoelectric module D15, and but in these studies the contact mode was used, and we see that even with working with pretty thick nanotubes, the probe still was ruining the structure. But after we applied hybrid PFM mode, we could run not only the same nanotubes but even smaller nanotubes with without distracting them. So on this slide, you can see the topography and lateral in-plane PFM phase of these diphenylalanine tubes. And you can see that there is a different polarization here. And also we could, together with PFM and morphology, we could also study the mechanical properties as we are running this same QNM at the same time. And it was shown that the Young's modulus of these tubes is around 30 gigapascals, which looks pretty close to the true. On the next slide, you can see the studies of TGS crystal. And again, we study, we have done some PFM studies, but you would say, okay, TGS crystal is pretty hard. Why not to use just standard PFM mode? The thing is that we changed we did the studies with changing the temperature in the AFM, and particularly in our AFM. It's pretty easy to change the conditions by changing the temperature of the sample. We have pretty large range from -30 to plus 300 Celsius, and doing in situ measurements of the sample. And in this case, you can see that the domain structure of the TGS changes dramatically going through the Curie point. So why not to use the standard good old PFM in this case? And this is explained on the next slide. We have done the studies what happens to the AFM tip when we heat the sample, because while heating the sample we also heat the PFM the AFM tip, and during the heating of course this tip starts to bend. And at this experiment here we were starting from 30 degrees Celsius and heating the sample up to 60 degrees. So this is this y-axis is time, and x-axis is temperature. And we were capturing at the same time the bending of the tip. And you see when we heat up for 30 degrees, tip bends up to 100 nanometers, which means that with the tip with force constant 0.5 nanometers, for 0.9 Newton per meter, we're getting 50 nanonewtons of parasitic bending, which is actually not acceptable. Tip will either detach, like it would be in this case, either start to scratch the sample. So, but as we are comparing the maximum force and the baseline in hybrid mode, we are adjusting the this set point automatically. It's done automatically in each measurement cycle. And at the same time, we can measure the PFM. And here from the left side, you can see the topography of the sample during the heating. Okay, there is some distortion, but it's due to the sample lateral drift, which obviously will happen when you heat something. And at the same time, collecting the PFM. So when the sample is under the Curie point, we see the domain structure. Then the domains become smaller, and then switch to the clear phase. So these in situ studies of even such easy material as TGS, but during the heating could be done only with this approach. So the next part of slides is dedicated to electrostatical studies of samples and electrostatical studies of smart materials. And electrostatical studies can be done if we have conductive tip. We place a sample on the conductive pack and excite the alternating voltage between tip and the sample looking at the response or at the electrical response of the tip to the sample electrical, which is close to sample electrical properties at different frequencies. And at different frequencies, using multiple lock-in amplifiers, we can, for example, map the surface potential at the first frequency, dc by dz at the second for harmonic, and dc by dv at the third harmonic, getting the electrostatical maps together with the morphology. And just one small example about these studies. It was done by the group from national from England, from NPL, and in this study, the graphene was measured. The work function of graphene was measured at different conditions and different relative humidity. And it was shown that the work function depends really much on what is the humidity, comparing like starting from 0, from vacuum, up to 60%. And it was shown that it changed really much at low values of humidity and goes to saturation only at about 40% of relative humidity. So that means that, first of all, if you make the electrostatical studies of such samples as graphene or other two-dimensional structures, you must take into consideration this humidity. And the second thing is that the devices which work on the basis of graphene or other materials of graphene-like materials should be encapsulated in real devices. So the next small example is about the magnetic force microscopy. So in magnetic force microscopy, we have the tip which has magnetic coating, which has magnetic moment, and if we are traveling along the surface with this tip, so that this tip feels the magnetic field or the gradient of the magnetic field, which is caused by different polarization of the sample. And we are capturing the phase which is proportional to this gradient. And in these studies, together with these, at least in our devices, we can apply and control precisely the external magnetic field in plane or out of plane. And in this study, the following thing was done. The change of magnetic polarization of these cobalt platinum nanotubes were done under the varying external magnetic field. So, and here you see on the top left, you see the topography, and at B, C, and D, you see how the magnetic polarization of these wires changed under application of different fields. So, as I mentioned, together with studying something, you can use AFM to modify your surface with a really high resolution. And in this cartoon, you can see the example of AFM oxidation lithography, local anodic oxidation. So, like I always was thought that nanolithography is something like a toy. Okay, you can draw something on the surface, show to your friends, but actually after that happened before we started to work with a team from Israel. They made the great work published in this paper in 2015, and this work was done on the nanolithography on OTS monolayer, and the OTS is hydrophobic, but if you apply some certain voltage, actually pretty small voltage, to locally, so these local areas become hydrophilic. So you can spend quite big time to make the template, nano template with the tiny figures on the stamp, and then multiply the stamp on other samples. So this is actually the first real application of nanolithography I've seen. And the second nice paper from German and Netherlands. This group was studying nanolithography of polypyrrole films, and it was shown that you can draw something with the tip with pretty small dimensions, but at the same time, applying different voltage, you can also erase the structures under certain conditions. So this is a really good beginning for AFM lithography application. So, and my next part will be dedicated to the combination of atomic force microscopy of scanning probe microscopy with optical techniques. So it's already like 21 years. 21 years ago, we have installed the first commercial system of combined AFM and Raman. Here on the slide, you see how nowadays, like our previous versions of the system look like. So from the right side, you have Raman spectrometer, which is combined with the AFM, either for on inverted microscope for transparent samples, or for opaque samples with the top view. And we have in these configurations we have different options for optical access and AFM access to the same spot, from top, from the bottom, and from the side. Also, we can use scanning tips with aperture, like SNOM tips with SNOM cantilevers, aperture cantilevers, or SNOM fibers. And on this slide, I show how the Integra Spectra, actually Integra Spectra 2, looks nowadays. So here we have improved the AFM performance really much, because it's very important to keep the tip and the sample in a good and stable contact, so without compromise of AFM part. And at the same time, we improved the optical access. So this is a pretty open design. So we can have access from the bottom, from the top, and from the side. And these objectives are really easy it's really easy to reconfigure your system, because all these objectives are on kinematic mounts, and you can just change objectives without much tuning. So one of the examples of application of this combination of AFM together with Raman is mapping of the graphene on the substrate. So from the top you see several images done by AFM, and at the same time we could capture the volume of spectra in each point exactly the same area of Raman. Getting the optical information from this. So the first good point is that you are working with the same sample at the same time, so we don't need to move the sample from one laboratory to another laboratory, finding all this, finding the place, and spending time for this. That's the first point, but it's not the exceptional points. You can live with this. Getting AFM together with Raman scattering together gives us sometimes a nonlinear effect, nonlinear value, which we call tip enhanced Raman scattering. So it works like that. When we bring tip to the laser spot position, under certain conditions, tip starts to work as an optical antenna, enhancing the Raman response from the area when the tip localized by the tip. Thus, we are getting two benefits. First of all, we enhance the Raman response. We reduce dramatically the collection time of Raman spectra, and the second and very unique advantage is that we break the diffraction limit resolution, and can collect the Raman spectra with the resolution which is much smaller than the half of the wavelength of the laser. And on this slide, you can see the scales here. It's 80 nanometers and 50 nanometers. So these are Raman maps of graphene, and we can see the defects, which are like tens of nanometers. And actually, this the number of TERS publications have grown starting from previous decade, and now we can see hundreds of them published, and most of them are done with our spectra system. Another way of combination of AFM together with optical technique is scattering SNOM in infrared range. So here is how our integral nano IR system looks like. We have the AFM part, and together with AFM, we have the detection based on the Michelson interferometer, and bringing the tip and detecting the nonlinear part of the tip oscillations. Here we can extract the information about the optical properties of the sample, which are actually defined by the size of the tip. That means that with the laser spot of like 10 micrometers, we can get the optical information of like tens of nanometers. And this is one of the nice works done by our customers from US. They were studying the so-called the metamaterial, which is also a smart material, of course. They were studying the phase change of vanadium dioxide, and they made the structure on the titanium oxide substrate, and this was chess desk structure, and they deposit the vanadium dioxide thin film on it, and on the surface of the structure the film had a regular structure, but in the bottoms it was unregular, and it had a tension. And when the vanadium dioxide was being heated, it was shown that the areas which were under tension started to conduct faster than areas which were well organized. And here on the slides you can see the maps of infrared maps done by IR scattering SNOM, which shows the intensity, which is proportional to the change of the dielectric permittivity, and the blue areas define the conductive regions, and the dark red areas show the insulating regions. And on this slide we can see more in detail how the mechanism of conductivity appears in this wonderful metamaterial with the heating. So at the room temperature, actually we see some conductive parts, which are also seen by scattering SNOM, and with the resolution which is much less than 1 micrometer and even less than 100 nanometers. With the laser spot, I remind is 10 micrometers. And after heating, these regions start to connect with each other, thus the whole film starts to conduct. And one more slide about the sensitivity of scattering SNOM in the infrared. And so from the left side we see the topography of ultra thin film of oligothetan, and from the right side we see that we see the IR SNOM map, and we see that each of these thin layers is clearly detectable with this technique, together with of course the higher lateral resolution. So the lateral resolution here is 1,000 times better than the wavelength of the laser. And finally, I just wanted to mention that most of techniques which can be applied to study the smart materials and constructive materials are implemented in all of our AFM systems. We start with pretty basic and finish with advanced and automated. So getting this AFM, any of these models, you are getting most of all, well, the whole set of these mentioned techniques. And of course, if you want to combine them with optical techniques, we have very specific and well working solutions. So finally, I want to refer to the papers I mentioned today, and after this webinar, you'll get the follow-up email, which will give you the link for the record of this webinar, and as well as today's presentation. And you can later on go through all these papers. And of course, I would like to thank all my colleagues which helped to get many of results presented today, and of course who are developing this wonderful hardware. And thank you for your attention. And Vyacheslav, I think we have some questions.
Thank you, Stanislav, for the lecture. We have several questions, and we have some time to answer some of them. The first question is there any possibility to control the bend of the tip of the cantilever while heating during the experiments where the sample temperature is varying?
Yes, thank you for the question. Yes, so the bending of the tip is you cannot control it without detaching the tip from the sample. So if you have the parasitic bending, in this case, you should take the tip away from the sample and measure this parasitic bending. And like I showed on one of the slides, when you did the heating experiment, the bending was pretty significant. And how we could avoid this? Just working in a hybrid mode, when we compare the set point of maximum bending and the baseline in every point, and subtract this parasitic bending automatically.
Thank you, Stanislav, for your answer. The next question: you've shown some examples of current measurements in different modes. What modes are available? In what modes is it possible to measure currents? And in general, is it just a contrast, or is it just a contrasts, or is it possible to make some quantitative results?
Okay, thank you. So regarding the ways how we do the conductivity measurements, so the standard way is go into contact mode and using the probe as a probe of ampermeter and the conductive probe and measure the conductivity in different points. At the same time, like I said, you can apply hybrid mode, which gives you advantage of measurement of complicated samples, which are not like concrete and stiff. And I have experience of doing this also in tapping mode. But in tapping mode, due to high frequencies we're working at, we're getting really small signal. So tapping mode can be used to measure the conductivity locally, but only at the materials which are highly conductive. Regarding the question of how quantitative we can go with this, actually it is not possible really to go really quantitative when you have only two contacts, like sample and the probe. But what you can do, you measure some quantitative numbers only when you apply at least like 4 probe method. But for this you can you need at least two additional probes. But then actually this was actually recently done, and we have integrated this probe station together with AFM, and soon we'll release it and show you how it works, and it's really very convenient. I don't know why we didn't do it earlier. So it will be possible now to do the quantitative studies.
Thank you. We have time for a couple of additional questions. The question is, can Scantronic be combined with still existing systems? So is it possible to probably have it as an upgrade?
Yes. The answer is yes and no. No means that the systems which have very old versions of controllers, they must be upgraded with the new version of the controller, which is like our common controller for nowadays. But the systems which run on the new controllers, all of the systems are compatible, and there is no need to change any hardware, any heads, or so actually any system, yes, can be upgraded with Scantronic, and no anything special in terms of hardware should be done. Okay.
Thank you. And probably the last one, we have several questions that are not answered, but I'll ask you one more question, and the remaining questions we will address through the email. Is it the possibility to measure morphology of a sample in solution?
Yes, yes, of course, it is possible, and we have like plenty of applications of AFM studies in solutions, because for most of biology, if you want to study the living biology, you must go to the solution, to at least to the buffer. If you want to study some in situ processes of like electrochemical reactions or deposition reactions on the surface, you must go to the solution. And of course, we have a pretty wide range of special liquid cells, starting from just petri dish, finishing with special holders for glass lenses. For eye lenses, contact lenses, yeah. So this variation is very wide, and there are different options to control the temperature, because especially for biological applications, some of your bacteria and cells, they live under certain temperature, like around 37 degrees. So, and there are also in some options you can change the solution during the experiment and see the in situ reaction of your objects to this change. Yes, of course, we do AFM. Also, we do even STM in the solution, and also we can do the combination of AFM and Raman in the solution. So yes, answer is definitely yes. So I think for today we will finish.
Thank you,
Yeah,
thank you, Stanislav. We still have several questions that we didn't answer because of the time limitations, but we will answer them shortly through the email. Thank you, Stanislav, once again.
Yes. So all of you will get the follow-up email with a link to this record and link to the presentation. And there is a small survey we prepared for you, and it will be very nice if you answer this after this meeting is finished. It will help us to improve our further performance. So thank you once again for attending. It was great, and see you on following online and offline events. Bye bye.